Okay, hello. Welcome to the Dissolved Oxygen Laboratory assignment. So the purpose of this lab is basically to see how the presence of certain types of microorganisms in water, like bacteria and stuff, affect how much oxygen is in the water. And oxygen is important for fish to survive in water and for us to have healthy legs. And so we'll then tie this into Minnesota. So what we're going to do, as you can see, we have a workstation here, and we have a nice timer. We have our yeast, which is going to act as our microorganism. We have water that we're going to mix the yeast in, so we're going to go ahead and do that. It will allow. And we're going to run 3 trials. We're going to use a little bit of chemistry here. Run 3 trials, and I'll be pausing midway through because sometimes there's a 2-minute wait for each one, and we'll want to go ahead and make sure that is good. So the first situation we have here, if we look at our actual lab assignment, we have no yeast. We're not going to use any yeast in phase 1, and our goal is to find out how much oxygen is actually in the water. So if you have a dissolved oxygen test kit for an aquarium or something, you may have done something like this in the past. For trial 2 or phase 2, we're going to have 2 drops of yeast, and for phase 3, we're going to have 4 drops of yeast. So we're going to start off with the first phase, which has no yeast. So what we'll do, we'll put in reagent A. 5 drops of reagent A. Shake it up. Five drops of reagent B. Shake it up. And then we have the timer. It goes for 2 minutes. And so I'll start the recording again when we get back to that point. Okay, so we've had our clock run down to 0. So what we're going to do now is we're going to add reagent C. And you don't need to know specifically what these reagents do at this point, because this is not chemistry class, but what we are doing is trying to figure out how much oxygen is actually in the water. And as you can see, it turned brown. So when it turns brown, or yellowish brown, that means that we are on the correct path here. And so when we add reagent D, the amount of drops that it takes us to turn back to blue color water will tell us how many parts per million there is of dissolved oxygen. So let's say it took 2 drops. That would mean that it would be 2 parts per million of dissolved oxygen. 5 drops, 5 parts per million. 15 drops, 15 parts per million. So take a quick guess here as to how many parts per million of dissolved oxygen there are. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 13 parts per million of dissolved oxygen, okay? So that was our total. So then we'd run, say, 13 parts per million. Awesome. Then we're going to go ahead and we're going to repeat the exact same thing with phase 2. And what I'm going to do is stop the recording again here, and then I'll restart it when we get to a point where we can continue. So what I'll do first, before I stop the recording, is I'll wash out the test tube and roll down this way, down the bench, and say, hey, all right. We're going to use 2 drops of yeast. All right. And we'll shut that and we'll shake it up and we'll run our experiment, and then I will return back when we're done with reagent C. The sample will be brown and we'll see how many parts per million the sample has. Okay, we are back. And so what we did is we had added 2 drops of yeast solution to this sample. And my question to you is, before we get going with this and see how much dissolved oxygen remains here, what do you think is going to happen to the amount of oxygen available in the water compared to our first sample? Our first sample was 13 parts per million. With the addition of this microorganism in here, what do you think is going to change? Do you think it's going to be higher? Do you think it's going to be lower? Okay, so let's go ahead and we'll see. 1, 2, 3, 4, 5. Remember when it turns blue. 6, 7. 7. Okay, so the concentration of our dissolved oxygen in the water is 7 parts per million. So what's the trend downward between yeast and no yeast? What happens when we add yeast? What happens to the amount of dissolved oxygen that's available in the water for fish when we add microorganisms like yeast? It goes down. And if we were to take this further, we would see that this is about 4 parts per million. But we don't need to because we see that this actually happens. So, all right. So yeast is a microorganism based on the data we collected. If there's a higher concentration or more microorganisms like a yeast or a bacteria in a lake, if there are more of those, how will that affect the lake's dissolved oxygen levels? The lake's oxygen levels will be lower because the microorganisms use the oxygen when they decompose other things, like algae, for instance. Okay, so the virtual lab showed us how to scale down into a small setting and experiment again in order to figure out what happens in the real world. And so as we move on, we're going to take a look at this graph. Lake Osakis is a nice place. You may have been there, you may not have. Does suffer from a bit of eutrophication, meaning algae blooms at certain times of the year. So we see this graph here, which has dissolved oxygen on the y-axis and month on the x-axis. So we're seeing how dissolved oxygen varies by month. In what month is dissolved oxygen the lowest? Correct, it is August. It is August. August is when the dissolved oxygen is the lowest. So the question is, when is the concentration of fertilizer the highest? So if we take a look at this here, nutrients from phosphorus and nitrogen fertilizers cause large amounts of algae to bloom, which is why you might see a lake that it looks green in the summertime. And the microorganisms feed on that algae once that algae dies, and that requires that microorganism to use oxygen when it feeds, just like when we need oxygen to metabolize food for our cells. So we take that from the atmosphere. And so, in what month then is the concentration of fertilizer the highest? It's kind of a tough question. It is August. Generally the peak of farming season, runoff is going into Lake Osakis. There's a lot of algae blooms going on, a lot of algae is dying, and it's being decomposed. And because of that, we have lower dissolved oxygen levels than we would otherwise. All right. So what we're going to do next, because we have about 7 minutes left of this recording, if we want to use all of it, is we're going to research a lake that's relatively close to your house. And in this case, we're just going to choose one. And I'm going to show you how to use the lake finder and what's interesting about it. So we'll go ahead and we'll do that. This is a great website from the Minnesota DNR. There's all sorts of awesome opportunities here. And so we're going to choose a nice little lake called Albert Lee, which is in Freeborn County, I believe. And we're going to take a look and see what its phosphorus content is. And so the DNR gives you all sorts of cool things about many, many lakes in the state of Minnesota, like for instance fish stocking, what is safe to consume for amounts of fish, water levels of the lake over time, fisheries survey, what types of fish are in the lake, water quality. We're going to focus on water quality. And we go to a page by the Minnesota Pollution Control Agency, who is in charge of monitoring this kind of stuff. So if you work for the PCA, you would be responsible for monitoring lakes for dissolved oxygen levels and how much phosphorus is in the lake and things like that. So if we take a look at Albert Lea Lake, and what area of the scale is Albert Lea Lake? We would call Albert Lea Lake eutrophic, extremely nutrient rich, shallower, greener. Then this is hyper-eutrophic. So basically, algae bloom is bad. It's green. You don't want to swim in it. Probably not very good for fish. Low dissolved oxygen, things like that. Total phosphorus in parts per billion, 252, which is pretty high considering that a lot of lakes generally average around 18 to 20 parts per billion of phosphorus. So, I'll answer a few questions here related to this, and then we will move on. Okay. For the last question, which type of landscape has lakes with the highest phosphorus nutrient levels, and are these fish healthy? Why or why not? So I made a bigger version of this, and so here is on the left-hand side, phosphorus content of certain lakes in parts per million, and then Minnesota land use on the right-hand side with a legend of what type of land use it is. So my question to you is, where is the highest levels of phosphorus or nutrients? In what lakes? In what parts of the state? What type of land use has the highest amount of phosphorus? Farmland, cultivated land. Cultivated farmland. There's a lot of nutrients from fertilizers, things like that go run off the fields into water sources, groundwater, things like that. That causes problems for a lot of different types of things. And of course, with the absence of dissolved oxygen, do you think these lakes have many fish? Yet they don't have many fish. And so the lakes down in the southwest and southern portions of the state are pretty eutrophic. They're in bad shape. And so really that's kind of the price that we pay when we farm. And obviously we need farming for food and things like that, and animals and all that. But there are side effects to that are environmental. And it's important that we understand how those work. So really, if you want a clean lake, head farther north. They're colder, they're clearer, there's less farming going on. And overall, generally nicer bodies of water. But that's just kind of the way that it goes. Same with cities. Cities can have a lot of phosphorus and nitrogens from fertilizers from people's lawns where that spews off. And so there are a few questions here at the end that I'll have you answer. But other than that, the goal of this lab was basically just to see how dissolved oxygen is affected by microorganisms in the water, and how we could make a controlled, scalable experiment in order to actually do that. So, thank you so much for watching, and have a wonderful rest of your day.